colleague. Okay. He's from the UK, as you'll soon tell by when he starts talking. Both got his BS and PhD University of Birmingham and University of Manchester. Then did a postdoc at Berkeley, where I worked in the lab with him for 3 years in Jill Banfield's lab. And then he was a staff research scientist at PNNL. And then he went to Ohio, and I knew that wasn't going to work out. So now he's at Colorado State, and he lives in the mountains, and he's a firefighter. Well, he's a volunteer firefighter.
Yes.
As you'll see, as his research has sort of led him into that direction.
Yeah,
So,
there's no escaping fire in Colorado, unfortunately.
Mike Wilkins, everyone.
All right. Well, hi, everybody. Thanks for coming out on a Monday afternoon, and thanks, Brett, for the invitation to come out. I've never been to Austin before, but I got in last night, had some good barbecue and some beer, and so it's all been going well since then. So today, as Brett mentioned, I will be telling you about some work we've been doing for the last couple of years regarding wildfire. As you'll see in my opening slides, wildfire is just a fact of life living in Colorado and much of the Western US, sadly, these days. But before I sort of dive into the main talk, of course, I want to let's get this going. Okay, all right. First technical challenges. Let's see. Wouldn't mind. It did work about 2 minutes ago, so.
Maybe there we go.
All right. Okay, well, like I say, before we dive into the data, I'd like to thank up front the people that have actually done the work, because I have very little role in this whatsoever. So pretty much everything I'm going to show you today was done by Amelia Nelson. She's a graduate student in my research group in soil and crop sciences. She's absolutely fantastic, as you'll see from the kind of data that she's generated over the last couple of years. This work is all reliant on access to these burned ecosystems, and that's primarily through the Forest Service. So we work closely with Dr. Chuck Rhodes, and he's a research biogeochemist in Fort Collins with the Forest Service. And we also try and, as much as we can, couple the microbiology with other data from these ecosystems, especially soil chemistry. And so we work with Professor Thomas Bork, he's a soil chemist, and Polly Roth is a graduate student in his research group. The work is supported through the National Science Foundation and USDA NEFA. And we have sort of instrumentation support through the Joint Genome Institute and EMSL, so user facility in Washington State. Now, as I'm sure many of you are aware, this is going to get really old if I have to do this every time.
I can do it. Oh, yeah.
Okay, so forest services in the Western US provide a whole range of ecosystem services. Globally, it's estimated that sort of enormous number of about $5 trillion worth of ecosystem services. And particularly in the Western US, you might have heard the phrase sort of water towers of the West. These sort of watersheds that you see in the background here, this is just outside Crystal Butte. These provide drinking water. It's the headwaters of the Rocky Mountains, provides drinking water to a huge population in the Western US and irrigation as well. And if you think about the carbon cycle, these sort of forested high alpine ecosystems store over 860 gigatons of carbon globally. And in the US, they actually play a pretty significant role in mitigating our CO2 emissions. It's estimated that about 40% of our emissions are offset by carbon sinks in these kinds of ecosystems. Now, within these ecosystems, I'm probably preaching to the converted here, but microbial communities are performing a lot of the processes that we're interested in. Okay. All right. So this is just an image from a nice review by Noah Farrar from a couple of years ago. And I'm not going to go through all the reactions here, but really the takeaway is that microbes are playing these key roles in generating gases, they're cycling carbon, they're cycling nitrogen, phosphorus, trace metals. All these processes in forests are catalyzed by the microbes in the soil. In addition to driving these sort of biogeochemical cycles, microbes are also critical for the establishment of the dominant vegetation types in these forests, which are pine, lodgepole, ponderosa. And so these kind of symbioses here, we just have image of fungal symbiosis with some pine seedlings, in particular ectomycorrhizal fungi, are critical for the establishment, persistence of these seedlings in these ecosystems. Now, like every ecosystem, these communities are susceptible to disturbance, and there is perhaps no greater disturbance than wildfire. So this is a photo that was taken just by CSU's campus in the fall of 2020. This was the Cameron Peak fire. And so I'm going to work and we're greeted by this sort of apocalyptic scene. Making this even more distressing is that, unfortunately, my house is about there on the map there. It didn't burn down, but as you can imagine, this wasn't the least stressful day of my life. So, so this was just one fire in 2020 in Fort Collins. And this really represents a trend that we saw that summer. So this is just a map outlining the sort of scars, all these fires that burned in this one fire season. So this is the Cameron Peak. This is that photo I just showed you. Obviously, here's where CSU's campus is. This was the largest fire. It recorded Colorado state history. But then we also had the East Charleston fire. I'm not sure if that might have made the news here. That burned into Rocky Mountain National Park. We had another huge fire up on the Colorado-Wyoming border, and then a series of other small fires throughout northern Colorado. So just a huge number of fires all occurring in the same fire season. And this really reflects this larger trend. The wildfires in Colorado and across much of the western US are becoming larger and numerous. So here we're just looking at acres burned and the number of fires. And here are some of those big 2020 fires. But if we plot burn area per fire and also total acres burned, you can see there's this positive trend through time. Okay. And this is just in Colorado. Now, traditionally, you might have thought we have this sort of very defined fire season, but even that seems to be not the case anymore. These are just a couple of pictures from the last 12 months, pretty much. So I'm sure this made the news. This was Louisville, just outside Boulder in Colorado. And this was December the 30th last year. This fire on a high wind day swept through that community and razed a huge number of houses. And then earlier this year, March 26th, there's actually snow up on the Flatirons in Boulder. And we still had this fire. This was called the NCAR fire. So these are both periods of time when we're not expecting to see wildfire and having to suit up and things like this, but we are seeing these fires seemingly year round. Finally, this is not just a Colorado issue either. Here it's just showing the trends in terms of area burned through time across these different sort of ecoregions. So we have the Southwest, Northern Mountains, and Western Mountains. And you can see in all of these different ecoregions, we're seeing this increase in areas burned by wildfire. And again, a whole variety of reasons behind this. Obviously, climate change is a big driver of this. We're seeing much drier forests, but also historic forest management practices as well. We haven't allowed wildfires to move through these forests. We haven't put enough money into forest thinning and things like that. So we just have these huge accumulations of fuels in these forests. So when they do ignite, we see a lot of fire. Now, this is important, a lot more moderate and high severity wildfire. Just to explain what you're looking at here. This is the burn severity map of the Cameron Peak fire, the one I showed you the photo of. And what I wanted to focus on are these areas of red. So this is high severity fire, and this is measured by the actual soil. The soil has been impacted by high severity fire. And traditionally, these forests are adapted to low severity fire and maybe even some moderate severity fire. A lot of the pine cones actually require fire to open and release the seeds. But these forests aren't adapted to this high severity fire. This is what you see if you look at bird landscapes and you see sort of those matchstick, completely combusted trees. And so we do see a lot of evidence of this in the Cameron Peak fire and many of those other fires that I highlighted a few slides back. And why is that important? Well, I'll just show you a picture here. This is just west of Colorado Springs. It's about 2 hours south of Fort Collins. And this was burned in 2002 by the Hayden fire. And originally, this was a ponderosa pine dominated landscape. And this photo was taken this year in 2022, 20 years later. And you can see that there's really no evidence of any pine regeneration there at all. You can see those matchstick trees. Okay, so that's the remains of those ponderosa pine. But for the most part, we can see they've been replaced with grasses, maybe a few shrubs. And so we have this potential as these ecosystems are burned by this high severity fire, these ecosystems are not adapted to. We have the potential to transition to sort of a new stable state of these ecosystems. So from forests to things like these grasslands and shrublands. Now, as a microbiologist who's interested in soil, I of course want to focus on what is the role of the soil microbiome in sort of facilitating these changes. So what does wildfire do to forest soils? Well, obviously, as you saw on that last image there, what does the fire burn? It burns the vegetation for the most part. So we of course see a loss of vegetation associated with wildfire. We also see a large increase in pH. If you measure ash in the immediate aftermath of wildfire, you can see soil pH measurements 10, 11, 12. So really high pH values following these disturbances. We also see the loss of secondary structures. So any sort of nice aggregates that you might have found in the unburned soils are usually completely gone. All the organic matter that's holding that soil together has been combusted. And so you're usually dealing with sort of very coarse, sort of sandy soils that really aren't very good at retaining water and things like that. And of course, any organic matter that hasn't been sort of fully combusted has been chemically changed for the most part. So we see some large scale chemical shifts in soil organic matter, and I'll briefly show you some data later on today. But critically, as a microbiologist, we're interested in this, the combustion of microorganisms. And this is a huge process that goes on during wildfire. Yamina Pressler had a nice sort of meta-analysis a few years ago looking at different fire impacted ecosystems around the world and came to the conclusion that fire reduces the biomass of microbes in soils by up to 96%. Okay, so in many instances, vast majority of microbial biomass in soils is lost due by this sort of fire disturbance. So with this sort of framework and this background in mind, we wanted to ask a couple of questions, and this is sort of how I'll show you the data today. First of all, how does fire change the composition of the soil microbiome? And after the loss of all that biomass, who do we see filling those newly available niches? When we're thinking about microbes that colonize this sort of burned soil, that's like a fairly extreme environment, which microbes do this and how do they do this? What traits do they encode that allow them to colonize these burned soils? Critically, when we're thinking about sort of ecosystem services provided by forests, how do these changes in the soil microbiome impact the function of the community? And then also importantly, how long do these changes last? Do these changes last for a year, or are we talking about decades? How long do we need to be sort of cognizant about these changes to the soil microbiome that are induced by wildfire? So I'll first direct you to this study that we've been doing the last couple of years. So we found two fires, the Ryan fire and the Badger Creek fire. These both burned in 2018, and they're located just up here in very northern Colorado on the Colorado-Wyoming border. And what we looked for were different burn severity gradients. So we wanted to see how does increasing wildfire severity affect the soil microbiome, affect function. So what we have here are 1, 2, 3, 4. These are our 4 different burn severity gradients. And we're going from a control unburned site to low, moderate, and high severity. And so this is just a graphic highlighting. This is actually a nice picture because often these gradients aren't quite as close as in this image here. But you can see we have the low severity region here where there's some slight charring and combustion of organic matter, all the way to a completely charred area here. So this was burned at high severity. All the organic matter on the forest floor has been combusted. And so hopefully that's fairly easy to see that quite clear gradient just over sort of 100 meters or so here. Often these gradients are slightly longer, trying to find the requisite burn severity. And just to get a bit closer to see what it actually looks like on the forest floor, of course, here's our healthy forest floor, our control and burn sites, low severity. So there's been a little bit of combustion, but you can see there's still a lot of organic matter on the forest floor surface there. Then we get into moderate severity. A lot of the organic matter has been consumed. And then high severity, you can see in the background, those trees that have been really severely burned, and really just ash on the surface of the soil now. So those are the changes in forest floor type and soil type as we go across this burn severity gradient. So we went out there and we collected both these sort of surface soils. So where there was an over-horizon, we collected the over-horizon. In many places like here, it had been replaced by ash. And then we also sampled deeper underground. We sampled sort of deeper mineral soils as well. And we did this about 9 months post-fire. It's sometimes very difficult to get onto these sites in the immediate aftermath of fire, just because of the risks of falling trees and
I guess you can't hear, right?
so we got there about 9 months into these samples. I wasn't quite sure what the audience makeup is going to be like, so I apologize if this is all too basic, but what we did with this was we sort of tried to couple some of the microbiology with the chemistry. So we sort of threw the microbial toolkit as it were. So we did screen all the samples with 16S and ITS sequencing. And then we also performed metagenomics to see what genes are actually present in these samples. And then we also used metatranscriptomics to see which genes are actually being expressed here as well. We tried to characterize the soil organic matter somehow. We chose this technique, a non-targeted technique called FTICRMS. It's a high-resolution mass spectrometry analysis that really gives you an inventory of the various compounds that are present in a given sample. So those were the analyses that we threw at these soils that we had we'd collected. So the first thing I want to highlight, and this is based purely off just the marker gene data, is that in the organic horizon, we see these very clear decreases in the diversity of the microbiome. And again, if we think back to that earlier slide where I talked about a 96% reduction in biomass, perhaps this isn't surprising in the slightest, but you can see here for both the bacterial and the fungal communities, and this is the surface organic horizon, as we go from control through low, moderate, and high severity, you can see that very clear decrease in the diversity of the microbiome for both of these groups. But then if we get down a little bit deeper, so into that sort of mineral horizon, we're not talking about meters down, we're talking about 10, 15 centimeters. But still, we're seeing a much more muted effect to wildfire here. There are still some decreases, but they're not nearly to the same extent as we see in the organic horizon for both the bacteria and the fungi. So it seems like deeper mineral soils, even though they're not massively deep on the ground, they do provide somewhat of a refugia after these disturbances. Now, in terms of the compositional changes that I mentioned, most microbes don't like fire, and that's what we can see here. What I've plotted here is the percent change with burns, so the percent change compared to those control samples that we collected. So you can see here that a lot of sort of common soil bacteria, so Acinetobacteria, the Microbial, they all decreased, often in sort of a dose-dependent manner with these increasing fire severities. So basically all the lines that fall beneath this 0 show decreases in relative abundance of these taxa following wildfire. Probably what will stand out here is that there are some microbes that actually seem to do far better in the aftermath of wildfire. So members of the Chloroflexi. Again, I'm going to highlight the Actinobacterium because perhaps unsurprisingly, they're going to keep coming to the forefront today as I sort of tell this story. And again, you can see here, they actually respond more strongly to more severe wildfire. So you can see the sort of dose-dependent response where we see the highest enrichment of these taxa with that high severity wildfire. Now, I sort of alluded to the fact this isn't particularly surprising because there have been other sort of compositional work, people studying the composition of the microbiome in other systems, whether it's boreal fires or fires in chaparral or even other wildfires in high alpine sort of montane ecosystems. And Actinobacteria have consistently emerged as one of sort of the positive responders to wildfire. Now, to start to get beyond these compositional shifts and actually start to understand a bit more about the microbiome and how it's responded to this wildfire, we started to use some of these other techniques. So we performed this sort of multi-omic analysis on samples from the surface, so the organic horizon and that mineral deeper horizon, from both low and high severity impacted regions. So this was the subset of samples that we selected. Obviously, I'm sure many of you recognize metagenomics and metatranscriptomics are sort of a lot more time consuming than using marker genes. And so while it would have been fantastic to throw these tools at every single sample, it wasn't really feasible with various sort of budget and time constraints. So we chose this subset. And of course, you have to have a sort of catchy acronym these days. And so we came up with the Fire Responding Ecogenomic Database, so FIRE-DB. And this accounted for nearly 640 of these microbial genomes. Okay, so we're taking metagenomic data and we're basically reconstructing microbial genomes. So these are these mags. We actually got 2 fungal genomes reconstructed, a lot of viral genomes, nearly 2,500 genomes of viruses from these same soils. And then, as I mentioned earlier, we used metatranscriptomics to look at gene expression as well. And this is just a breakdown of what these genomes that we reconstructed look like. And luckily, as you can see, we sort of expected to see a lot of actinobacteria genomes, and sure enough, those were the majority of the genomes we reconstructed from this omics data set. So we were sort of fairly happy that our MAG database here, our FireDB, sort of represented what we saw from the 16S marker gene data set I showed on the previous slide. So what are the critical traits that allow things like actinobacteria to do such a good job of colonizing this sort of extreme environment after wildfire? High pH, loss of secondary structure, completely altered soil organic carbon profile. Well, we can actually think about this using other systems that have been well studied post-fire. And these are, we can look to sort of work done on the ecology of vegetation. And this is a diagram that I have stolen from Dylan Enright's work. He's a grad student in Sydney Glassman's lab at UC Riverside. And when it comes to vegetation responses post-fire, ecologists have sort of grouped vegetation into these three different sort of guilds almost. You have vegetation of stress tolerance. And so, for example, we're talking about vegetation that produces structures like pine cones that only open in response to fire. So heat activated germination. Alternatively, you might get some vegetation that's well adapted to use resources in the aftermath of fire. So the ability to break down sort of pyrogenic organic matter or something like that. And then if you've been in a burn scar, you will often see vegetation like dandelions and thistles. And these are, this is vegetation that's able to quickly colonize this sort of empty space, this open space. So rapid expansion, analogous to resprouting and rapid colonization. And so with this framework in mind, we can think about maybe the microbes may exhibit some of these same traits. Critically, what happens here, though, in terms of the vegetation, is that these traits trade off against one another. Okay, so if you produce thermotolerant structures like pine cones that rely on heat-activated germination, you're also not growing quickly like a thistle or a dandelion. Okay, so you sort of choose one or the other trait to allow you to make the most of this ecosystem. So the question is, do these same principles apply to the soil microbiome after wildfire? We'll just quickly take a look at thermal tolerance mechanisms, so analogous to like heat-resistant structures, heat-resistant germination. So it's slightly confusing. I'll just explain what you're looking at here. We're just looking at the change in relative abundance as we go from a low severity sample to a high severity sample in both the mineral and organic layers. And so really what to take away here is that as we go from low severity to high severity, we see a big enrichment in genes associated with sporulation in both the organic layer and the mineral layer. And the same goes for heat shock proteins as well. So these are proteins that help stabilize other proteins and cellular structures within the bacterial cell. So it seems like whoever is being enriched in these post-fire ecosystems is able to utilize strategies for thermal tolerance. Okay, so that's one potential mechanism. First mechanism, the ability to persist through disturbance via thermal resistance. Seems to be a thumbs up. Okay. We next wanted to look at this idea of fast colonization. So analogous to this would be thistles and dandelions growing in those burn scars. And so here we used a tool out of Jed Furman's lab. And here we used computational tools to estimate the maximum doubling time of microbes. And there are certain signatures present in microbial genomes. It's called codon usage bias. It allows us to take those genomes that we reconstructed and actually estimate the maximum doubling time. And this was a nice figure from that graph where they actually calculated these doubling times from the microbial genomes, and they actually have the corresponding microbial isolates in culture in the lab. And so they actually observed the growth rate in culture in the lab, and they actually calculated it, the growth rate from the genome. And so you can see that while it's not a perfect correlation, it's a pretty good correlation. And so this gives us some confidence moving forward that we can take these growth rates that we infer from microbial genomes, and they'll be based somewhat in reality. So what do we see? Again, I'll just walk you through what you're looking at here. So we have on the bottom here is average relative abundance. So from 0% relative abundance to 6% relative abundance. And these are those surface soils, and these are surface soils. So this is high severity surface soils, and this is low severity surface soils. And hopefully what you'll see here, especially in the high severity condition, is that in general, the taxa that are the highest abundance, 9 months post-fire, remember, that's when we sampled, also have some of the highest predicted maximum doubling times. Okay. That signal still exists after low severity fire, just not quite to the same extent as after high severity fire. So now let's look at those deeper mineral soils that maybe weren't disturbed quite as much to see if that signal persists even deeper. So again, we're looking at high severity and low severity conditions just deeper down. And hopefully it's easy to see there that that trend is not apparent in those deeper soils here. So what's the sort of the takeaway here is that we have taxa, especially in these surface soils. If you think about all that biomass that's been combusted, 96% loss of biomass, we have all that open niche space for something to take advantage of. In the plant world, it's thistles, it's dandelions. It seems like certain microbes can do the same thing. In the aftermath of wildfire, microbes that can potentially grow the fastest do that and then account for a larger fraction of the microbial community up to 9 months post-fire. So a second trait that helps colonizing these soils is this ability to rapidly fill these unoccupied niches by using the potential for fast growth. Let's just take a quick deviation into some soil chemistry here, because this will be important when we think about microbes actually using soil organic matter. So here we just have an image showing just an extraction here. So we simply take mineral soil or ash that we recovered from these sites, and we do a very simple water extraction. That's what you're looking at here. And then we used this high resolution mass spectrometry technique, FTICRMS, at Emsl in Washington, to actually analyze the composition of these extracts. So we're looking at water soluble molecules. These are the kind of molecules that the microbes potentially could be using. Now, if any of you have used FTICRMS, you'll be sort of familiar maybe with these Van Crevelen diagrams. Each circle here just displays a different compound that was detected by the mass spectrometer. And we plot it based on the oxygen to carbon and hydrogen to carbon ratios, just to show the distribution of these molecules. And here what I'm showing are the molecules that are unique to each of those conditions. So molecules unique to the control and burn soils, molecules unique to the low severity soils, and then molecules unique to both moderate and high, which we grouped together for the purposes of this diagram here. Now, I realize this is sort of quite a messy figure with a lot of overlapping data points. But hopefully, if you squint a little bit, you will see that the vast majority of these red data points that are from the moderate and high severity fire group below this line that is basically where aromatic compounds fall for the most part. And you'll see other compounds, especially in gray, a lot of those and the yellow ones fall above this aliphatic line here. Just as a different way of displaying this, maybe a bit more clearly to you all, you can also take each of these compounds and calculate what's called the aromaticity index. It's basically a measure of how aromatic that compound is. And so this is a lot more clear. Make the slides work. So we have a low aromaticity index here in our control extracts. And again, this sort of very clear trend as we get to increasing wildfire severities, that aromaticity index increases. So basically, is it rocket science? Just the carbon that's left in these soils is more aromatic, which is what we'd expect after fire. You generate a lot of these aromatic structures in pyrogenic organic matter. So the question at this point is, can the microbes use that aromatic carbon that's been generated? We know we've generated a lot of this stuff. Are the microbes touching it, or are they leaving it alone? Now, the problem with assessing this, as you saw in the previous slide, there are a lot of different compounds made in sort of this pyrogenic organic matter. Lots of different aromatic rings stuck together with different side structures. And it's very challenging to look at a microbial genome and decipher, can a microbe break apart that sort of compound with 17 aromatic rings? What we do know, however, is that the aerobic processing pathways for a lot of this aromatic carbon progresses through two compounds. And again, I'm simplifying things here. These aren't the only pathways. This is just good for our purposes today. But protocatacoates and catechol are two compounds that are common intermediates in the processing of a lot of these more complex aromatic structures. And so for the purposes of this work, we wanted to see, can the microbes process catechol and protocatechol? We're assuming that a lot of this sort of more complex stuff is being funneled through these intermediates. And the answer is yes. So don't want to get too technical here, but these are the reconstructed pathways for the processing catechol, protocatechol, and eventually there's the potential to take these to TCA cycle intermediates, so straight into central metabolism for these microbes. And this is based just on looking at the genome. So all these genes are present in our soil microbiomes. Okay, so the genomic potential is there for the processing of these more complex aromatic structures, all the way to very simple energy sources. Of course, it's one thing knowing the genes are present, it's another thing seeing if they're actually being expressed in these samples. So what we did was we took that metatranscriptomics data, so the messenger RNA that we sequenced, and we mapped that onto our metagenomic data, into our MAGs. So I'm going to show you the same graph, but just with that gene transcript data. Here we're looking at, under high severity conditions, surface and slightly deeper A horizon, mineral soil. And what you can see here is that for the most part, these genes were expressed in every condition. And the thickness of the arrow just gives you some idea, some indication of the extent of expression, which isn't super important right now. But the takeaway is that not only were these genes present, they were also being expressed in these soils. So the microbes there are actively taking catechol and protocatechol and generating these compounds here. I just want to take one slight dive a little bit deeper. We can actually look here, and I apologize, this is kind of small down here, but each of these names, these are individual genomes we've reconstructed. These ones are dominant in the surface, O horizon. These ones are dominant in the mineral soil, okay, in the high severity fire. And the details don't really matter here. What I want you to take away is that whilst a lot of these dominant MAGs contained good portions of the pathway, so here we're going all the way from Cat A up here, PCAF, which is here. So whilst a lot of these MAGs contain a sizable fraction of these pathways for degrading this organic matter, none of these MAGs actually contain the entire pathway. So takeaway here is that these microbes are working on the organic matter, but there's likely a lot of sort of metabolic handoffs of these compounds. This is likely a sort of a cooperative effort between different soil microbes that allow these compounds to be taken all the way to succinyl and acetyl CoA. So coming back to this framework, our third trait was the ability to generate pyrogenic carbon. And based off our genomic and transcriptomic data, it certainly looks like a lot of the dominant microbes, a lot of the dominant actinobacteria in these systems can do this as well. Now, if you remember when we were looking at that conceptual figure, I made the point that we expect to see trade-offs when we sort of consider vegetation. Again, the vegetation producing pine cones aren't the same things that are growing fast like thistles. They trade off against one another. But instead, when we look at this through a microbial lens, what we see is that many microbes can use all three of these traits. There's no real trade-offs from what we can see. So things like actinobacteria that we see just doing incredibly well in these burned soils, we think can do this because they have thermal resistance mechanisms. They produce spores. They can grow fast when there's all these unoccupied niches in the soil. And then finally, they can also use the most abundant carbon sources present in this system. So they're really ideally placed to colonize these systems. And that accounts for that real enrichment that we see of those microbes up to 9 months post-fire. I just want to talk briefly about viruses. This probably isn't the greatest data set to study viruses, but I thought I'd mention this nonetheless. So viral activity, as I'm sure many of you can know, has the potential to contribute to carbon cycling as well. And this is through this mechanism of sort of top-down control here. So basically through the viral lysis of microbial cells, and then of course we have viruses, and all that sort of tasty organic matter that is present inside the cell can leak out into the ecosystem, and that can represent another very abundant carbon source in many ecosystems. It's mostly been studied in marine systems, but there's no reason to think these same kind of processes can't happen in sort of terrestrial soils. Now, I mentioned at the start that we were able to reconstruct a lot of viruses in the same way that we reconstructed those bacterial and fungal genomes. We also reconstructed these viral genomes. About 2,500 distinct soil viruses were discovered across this data set. For those of you that work on viruses, I say 1,000 of these viruses are novel. Usually, whatever ecosystem you look in these days, you discover a lot of viral novelty. It just hasn't been explored that extensively yet. So a lot of viruses and a lot of viral novelty. But what we wanted to do was basically try and work out which bacteria are these viruses targeting. These viruses need to invade bacteria to replicate and destroy those cells. So I'm going to show you a very similar graph to one you've seen before. Again, we have relative abundance, so 0 to 6%. Again, these are surface soils impacted by high severity wildfire. And what you can see here is that we've calculated the number of viruses that we can link with all the different bacteria. The takeaway here is that the viruses that are most abundant, so up here, also generally have the highest number of viral linkages. And interestingly, it's the same. We go into some slightly deeper soils, the A horizon impacted by high severity wildfire. The most abundant mags also have the highest number of viral linkages. And so this kind of data reflects this strategy called kill the winner. Basically, the viruses target the microbes that are doing the most successful in a given ecosystem. Sort of predator-prey dynamics, if you think about this in sort of a macroecology sense. But the fact that we're seeing the most abundant bacteria being targeted by viruses suggests that viral predation is an active process going on in the system. So in addition to that degradation of pyrogenic carbon, I think we can also expect in these soils that a lot of labile carbon is also being turned over by the lysis of these microbial cells, a lot of these actinobacteria that are highlighted in blue here. Now, I've spent the last sort of 15, 20 minutes talking to you about how certain microbes are able to colonize these systems. And I haven't really said much about what's missing. So just briefly, this is just as important. What vital ecosystems are missing 9 months after wildfire? When we talked about that sort of $5 trillion number for ecosystem services provided by these watersheds. So what have we lost in the aftermath of fire? This is probably one of the more critical ones. I showed you earlier on, especially in those superficial soils impacted by high severity wildfire, there's huge decreases in fungal diversity. Now, critically, a lot of that decrease in diversity can be accounted for by the loss of ectomycorrhizal fungi. And we see 9 months after wildfire, the almost complete loss of ectomycorrhizal fungi. And these are those critical symbionts of lodgepole pine establishment and growth in these soils. So of course, that has implications for the regeneration of these systems if we expect these seeds to germinate and these trees to sprout back up. I haven't talked much about the nitrogen cycle, but wildfire does alter the nitrogen cycle massively. We see big inputs of ammonium into the system, which can be nitrified, it can be leached out. So in the aftermath of wildfire, the ability to fix atmospheric nitrogen could potentially be really important in adding nitrogen back into these highly disturbed soils. Unfortunately, we didn't see any evidence of nitrogen fixation going on in these soils at all. We were able to find some NIF genes, but there was no evidence of these NIF genes actually being expressed at all. And so it's clear that we do have a significantly altered nitrogen cycle in these soils at this point in time. And then finally, the stability of pyrogenic carbon. Pyrogenic carbon, these sort of highly aromatic structures, are traditionally thought to be really recalcitrant to microbial degradation. And yet we show here that the dominant microbes in these systems are able to process this. They're actually actively processing this at the time of sampling. So this definitely has some implications for us of understanding of post-fire carbon storage after these disturbance events. Now, just if anyone's interested in this stuff, this just came out about a week or two ago. So if you want any more information on that, it's in that paper. This work actually got on the cover of Nature Microbiology. I snapped that photo with my cell phone, just on a whim in the forest. And then, 2 years later, it's on the cover of the journal. So you don't need those fancy cameras. Okay, so probably about 10 minutes left maybe. So I will just finish up today addressing one of the questions. This work all focuses 9 months post-wildfire. It's a snapshot in time. One of the questions I posed at the start was, how long do these changes persist for? So we'll get into that now. Now, if any of you have walked through forests in the Western US, I'm sure you've seen these structures, these sort of slash piles. So as forests are thinned or clear cut occurs, and there's sort of leftover slash material, it's piled up into these big structures. And then when it's cold in winter and there's very low airspeed, very low wind speed, these things are set on fire. It's a fuels reduction treatment, and very effective at getting rid of fuels in these forests. But they leave this legacy scar. So this is just an aerial satellite image of the forest in North Park, Colorado. And there are these clearings in the forest. And of course, they don't look completely circular, but when we get down here, this is a photo of what was previously a clear cut. These trees have all grown back, probably about 40, 50 years of growth there. And this is the area where one of those pile burns was placed and was combusted. And you can see that whilst outside the pile burn, these trees have all grown back after being clear cut. Now, decades later, really, apart from a few small little plants, nothing substantial has grown back here in this burn scar. Okay, so we have these legacy scars scattered across forests across the Western US. And the way we're using these scars and these pile burns is as a proxy for severe wildfire. So we have this large amount of fuel, burns the ground severely, doesn't move fast, obviously, because it's a static pile of fuels. So we're using them as this proxy for very severe wildfire, and seeing what happens to the soil microbiome after these events. And this is really enabled because the US Forest Service in North Park has kept really good records of when they burned these pile burns. Often the records aren't that great, and we just find the scars in the forest with no real indication as to when the pile burns were burned. But here in North Park, we know when piles were burned in the 60s, 70s, 80s, 90s, and 2000s. We have the GPS locations of these scars. So this acts as a really nice chronosequence for really high severity wildfire in this area that really isn't, it's a fairly small area. So we're controlling for elevation. We're generally controlling for aspect. We're controlling for fuel type. So a lot of the problems that you might associate with chronosequences, we can actually control for with this experimental design. So what we've done is we've selected a large number of scars across these decades, and we've gone in there, we've collected surface soils and analyzed them with 16S ribosomal RNA and ITS, so marker gene sequencing, and then again, doing some metagenomic sequencing. I'll show you a little bit of data from a slightly unique aspect here. We're actually able to take lodgepole pine seedlings and plant them in these different scars for 3 years and see how over that 3-year period those lodgepole pines, they survived or not, and also what kind of rhizosphere communities they recruited over that 3-year period in these different soils. So this really is to test the question, what's the timescale of soil microbiome recovery following severe wildfire? Have this chronosequence going back to the 1960s. So I'll just show you some marker gene data now. These are simply multivariate statistical analyses, so NMDS analyses. And what we're plotting here are soil microbiome in the burn samples in red, and then the unburned controls in gray. In the 2000s, 90s, 80s, 70s, and 60s scars. Now, I don't want you to have to squint your eyes too hard, but the takeaway here is that basically we don't see convergence of the communities between samples taken within the burn scars and samples taken outside of the burn scars until we get to the 1960s. So if we're thinking about this from a purely compositional sense, we really don't see convergence of these communities for about 50, 60 years post-wildfire. That's how long it takes for the communities to start to look similar to each other, outside the burn scars and inside the burn scars. But remember, this is just composition. So does the community look like the unburned community? The other question to ask is, well, just certain functions come back sooner than that. Okay, just carried out by different microbes. So I'll just want to focus on carbon cycling for the next slide or two and just explain what you're looking at. Again, we have this chronosequence here. So this has had the longest time to potentially recover, and these are the most recently burnt piles. What we're doing is that we're summing, this is sort of a relative abundance calculation of genes associated with a given process. Here we have the unburned samples and the burned samples. So polymers, linear hydrocarbons like alkanes, are almost always more abundant. Genes for the degradation of these compounds are more abundant in the unburned samples relative to the burned samples. And it's only in the 60s that we start to see that kind of functionality return in those unburned samples. But we can completely contrast this with the processing of aromatic carbon. Hopefully those slides weren't too long ago, and you remember that we do see this big enrichment in aromatic structures following wildfire. And the ability to the genomic potential for the degradation of these aromatic structures is actually greater all the way through to our last sort of point in the chronosequence. So the potential for aromatic carbon processing is always more elevated within these burn scars, and at no point does it ever look like our control samples. And then interestingly, we also look at sort of more complex polyaromatics, and we see a difference sort of 20 years ago in the 2000s. And then after that, the data actually tracks pretty well. So obviously, as the soils are working in, there's definitely some noise in these systems, and so the data isn't always as beautiful as we'd like it to be. One other carbon processing enzyme are polysaccharide lyases. So remember, 96% of biomass is degraded, is killed by wildfire. And so enzymes that help to process like necromass, dead biomass, could be potentially useful in these systems. And sure enough, we see in the 2000s and the burn piles in the 90s, much more abundant enzymes within these polysaccharide lyases compared to our unburned controls. And again, once we get into the 80s, we start to see some convergence there. So the takeaway here is that while the community takes about 50, 60 years to converge in terms of community composition, if we're thinking about different functions within the soil microbiome, there's a lot more variability. Here, in the 80s, we see some level of convergence. Here, we see convergence in the 90s. So it's not such a simple story as the soil microbiome takes 60 years to recover. A lot of those functions are retained more quickly. Other functions are potentially more enriched, such as like benzene degradation. And there are other functions that are lost forever. Or at least to the end of our chronosequence, I should say. I want to just revisit the nitrogen cycle because I mentioned that the nitrogen cycle undergoes like a huge disturbance. Denitrification is massively enriched in our burn samples, all like multiple decades until the 70s. So I haven't quite fully thought through this. Is fairly new data. Haven't quite got a good explanation for this, but again, this just really gets at the point that the nitrogen cycle is massively altered. And the same goes for nitrogen fixation. We saw 9 months post-fire, there was really no evidence of nitrogen fixation occurring. But luckily, at some point, maybe 1 year, 2 years, 3 years post-fire, nitrogen fixation obviously does kick in because we see an enrichment of nitrogen fixation genes in the 2000s. And then after that, it maps pretty well to the unburned control samples. Okay. So the nitrogen cycle is something definitely that requires more digging in some of these stories. I just want to finish up briefly with a quick mention on some of these seedlings that we planted. So the seedlings look pretty much like this, and we were able to plant them within these burn scars. And we pulled them up when we sampled all these soils, and we sampled the soil associated with the roots, and we performed 16S and ITS sequencing to see what those rhizosphere communities look like. Again, I'm not going to show you any compositional data. I'm just going to show you the diversity of the communities in bulk soils and in the rhizosphere. So the red is the burned bulk soil from the burn scar, and the green is the soil that was associated with those lodgepole pine seedling roots. And you can see that for the first sort of 2 decades, we really don't see any difference between, in the diversity at least, of the soil between the roots and the burn scars. But then starting in the 80s, 70s, and 60s, in these soils that have had longer to potentially recover, we start to see evidence of maybe selection. So the actual plant is hopefully trying to recruit beneficial members to its rhizosphere. And so we start to see that here, here, and here, with this reduction in the diversity of the microbiome in these plant roots. And we're taking this as evidence that the plant is actually actively able to select certain beneficial microbes in these older soils that have had longer periods of time to recover. And just to mention our ectomycorrhizal fungi, I talked about how 1 year or 9 months post-fire, we saw this sort of almost complete wipeout of EMF. And even on the decadal timescale, we can see that EMF are significantly depleted 20 years ago and 30 years post-fire. And it's really only in the 80s, 70s, and the 60s that we see a rebound in ectomycorrhizal populations. Unfortunately, if there aren't EMF occupying this niche space around the lodgepole pine roots, other things will take advantage of that open niche space. And indeed, we see fungal plant pathogens, higher diversity in these rhizosphere samples. And then as the EMF come back, we see a decrease in the abundance of the plant pathogens. And so EMF are essential for providing elements, nutrients to the tree, but they also are essential because they occupy this niche space and prevent these plant pathogens from occupying that space. So 2 roles for the EMF. It's one reason why they're so important in these burn soils. So this doesn't have a nice bow on it. This is like very recent data that's just been provided to me. So I haven't got a nice tagline for the end here, but to just sort of start to wrap things up. Hopefully, I've convinced you today that increasing severity of wildfires, so these increasingly severe wildfires that we're starting to see out in the Western US, are driving larger decreases in just the diversity of the soil microbiome. I mentioned that actinobacteria was going to be a consistent theme today. They do a very good job of colonizing these soils because they encode multiple traits. We saw how they can grow fast, they can potentially survive a lot of the soil heating, and they can also use the abundant altered pyrogenic carbon that's generated. Viruses, always an area that need more research, but it certainly looks as though viral abundances and viral predation likely plays a key role in this ecosystem as well. And again, that may be able to supplement sort of nice labile organic carbon through the lysis of the microbes that are living in these soils. EMF, the ectomycorrhizal fungi, is likely a critical loss, especially after increasingly severe fires. Truly got to the bottom of if that's the main reason why we don't see regeneration in burn scars like the Hayman burn scar. But it's reassuring to see that at least in those burn piles, we were seeing rebound of EMF, albeit on sort of 40, 50, 60-year timescales. And again, this gets to that point that the timescales that we're talking about for recovery of the soil microbiome and soil microbiome functions doesn't happen over months or years. We need to think about this as changes that happen over decades, even potentially up to sort of 60 years after fire, until communities start to look similar to one another. So I think I'm making about 5 minutes to 5, so hopefully I didn't go too long. But thank you all for coming today and listening, and I'll take your questions if you have any. Questions? Maisie?
Yeah, thanks. That was amazing. I wonder, so I think all your 16 sample kind of stuff was all just relative numbers. Yeah. So do you ever try to get absolute numbers?
Yeah, we've done some quantitative PCR to get numbers. The problem is the biomass is pretty low in these systems after fire, and so a lot of the data is frequently very noisy. I explicitly didn't show you any qPCR data today just because the error bars are large between our replicates and things like that, and so it's kind of unsatisfying. But we're also playing around with lipid analysis now as well to try and get a measure of overall soil biomass and things like that.
But it goes down.
Yes, yeah, we see those same reductions.
So this thing with the actinobacteria, I mean, might not be so much that they go up, they just don't go down as much.
Yeah, they may just be survivors. I mean, yeah, see an increase in the relative abundance, but if your biomass has gone down, they're just better at persisting through that spore formation. like I say, I think the fact that they do show those rates of fast growth suggests that they are somehow exploiting those niches. Once the ground is cooled and they're able to grow again.
Well, go ahead.
Yeah, I'd like to talk recently about soil engineering, if you will.
Yeah.
Are there efforts like that covering those fires?
There's on the microbial side of things, not really. Sometimes people will dip seeds in like EMF and things like that to try and enhance the germination. I think probably in terms of like engineering these systems, people just go out with seedlings and just plant them. Some people have these bags of seedlings in a small shovel and they can plant like 1,000 trees a day just hiking across these landscapes. So after the Hayman fire, there was a lot of issues with reservoirs, water supply to Denver. The reservoirs was just like burned hillsides all around them. There was potential for a lot of ash runoff and things into those reservoirs and compromising water supplies. And so Denver Water spent like millions and millions of dollars just basically because the trees weren't going to grow back, they just planted them there, kind of artificially recreate the forest after the Hayman fire. And it works. It's just it's not potentially not sustainable on the kind of scales that we're seeing these fires occur now.
My question was kind of similar to that one. So do you see potential for bioremediation? Like you know what are the microorganisms that are responding in like high this fire soils? Like is there a way in which you can treat those soils with the microorganisms that are gonna help those seeds to regrow?
Yeah.
What's the question?
Can you sort of do like a bioremediation approach here and actually amend the landscape with beneficial microbes that would provide the services you need? I think the problem that I think potentially you could do, I think sort of something that they're doing now, a more tractable solution is change the soil conditions so that it's more amenable to microbes to grow. So what they're doing on Cameron Peak fire, if you drive up west of town on a weekend or any day of the week, they're flying helicopters and they contain huge containers of mulch, just like the kind of mulch you put on your garden. And they're dropping mulch on the landscape to try and prevent erosion. It helps to retain soil moisture. It stops the surface soil getting really hot in the summer. So this mulch basically creates conditions that are more amenable for hopefully microbial growth to try and sort of rebound, and we'll see that increase in biomass. So that seems to be a more likely strategy than sort of spreading microbes around, I think in the short term at least.
We recently had a fire in our fuel station that's in a pine forest here. And one thing that was noticeable there, and I'm sure it's true because not in your first slides you have a total wipeout, but ours is more of a mosaic, right? You have everything from really hot fire to just a light understory fire. The thing is that we really didn't have a baseline of the microbial community. And what I'm wondering is whether just taking what didn't burn and comparing what did burn is really fair, because it doesn't seem like it's random. Some of the places that were really hot fires might have had a pre-existing difference from the areas that didn't burn hot. And so, without the baseline in the hot fire area, you're not sure if you've really measured the transition.
Yeah, I mean, it's definitely a challenge with fire. You never really know where it's going to. I mean, the only way you can get around is with prescribed fire. If you know there's going to be a prescribed fire, you can take those before fire samples and obviously the post ones. But yeah, that's a challenge. I think you're right. The areas that burn more severely, there may have been more fuels on the ground, which of course, affects some soil conditions and whatnot. So I think what you can do is just sort of increase the number of samples. If you were going to do a study like this, try and sort of take as many samples as possible to try and account for that sort of inherent heterogeneity in forest soils. And of course, the data is going to be noisy, but hopefully if a sufficient number of samples are taken, you start to get at some of those sort of underlying differences at that stage. But no, I totally agree that there are a whole range of sort of forest factors that affect where you sort of see these variation in fire severity.
The other question, I'm sorry, the other question has to do with the depth of your sampling.
Yeah.
And so, you have a root zone much lower that would provide a source population to the surface.
Yeah, that's something we're working on right now, trying to work out like where do the microbes come from after the surface has been cooked. Are they coming in as sort of spores in the atmosphere? Are they migrating from deeper? Yeah, there's right.
Well, I had a related question actually, but did you collect any of like plant performance data? You know, you talked about the pattern of reducing richness, making signature selection. Like, did those plants do better in the seedlings?
Yeah, we did. Our Forest Service collaborators were out there measuring like stem thickness and things like that, and tree height and whatnot. Actually, that's something that we're still working on right now. I can't tell you whether there was like a statistically significant. They were doing better in those older bird scars. To the eye, it doesn't look like it. The trees stay alive. There's no real evidence that they're growing that happily. They just kind of cling to life. But again, my eye is the same as some of these nice measurements they took. So I'll have to get back to you on that.
Okay, one more. This is from a grad student.
Thanks for the talk. This is fascinating. And especially I think it's really cool that you're testing basically if there are trade-offs between the stress tolerance being brutal and being competitive. And actually, I think in the past several years, there's like a conceptual article that's basically saying there's the YAS. Yeah. So I think it's super cool. And one thing I was, so basically you're doing the comparison of the microbiome within the burn site, within like basically a system that has frequent disturbance, which is the fire. And I wonder if you have like done comparisons of basically with other systems that's not that has like similar vegetation types but does not have a fire, for example. And maybe like comparing the expected growth rate of the microbiomes in Colorado versus in the other place where they it's not like they do not experience as much disturbance. So I wonder if there's like a comparison outside the system and see if that trade-off will show up.
Yeah, that's a great point. I'm trying to think. I'm sure we must have calculated, done those estimated maximum growth rates in our unburned controlled samples. I don't know why I haven't shown that. We must have that data somewhere. So I need to dig that out and just make sure. I'm hoping it doesn't show. I mean, you sort of saw that in the mineral horizon. So the deeper soils didn't show that same sort of correlation between relative abundance and growth rate. So my assumption would be that in the unburned samples, where the diversity is higher, we don't see that correlation. But I must have that data somewhere. I just need to follow up, I think, make sure that's the case. And one thing to say though that I didn't have time to throw in today is that when we're studying these burn piles, we also see that same correlation, that evidence of like fast growth rate is beneficial in the microbiomes, in the microbes that colonize the burn piles as well. So that does seem to be a consistent response to both natural wildfire and also these kind of artificial proxies that we're using as well.
Thank you.
All right. Thanks, Mike. That was great.
All right.
I have a lot of questions, but I